This is the second part of the two lectures about using DNA for forensic purposes. The first one dealt with what DNA was and what it looked like and the biological implications of DNA. This is going to mostly deal with the forensic use of DNA. Once you know what it was from the other lecture, now you can see how it's used in forensics. The basic idea is to get first a sample of DNA, then you replicate the areas of the DNA. These are the STRs, the short tandem repeats. We'll talk about this soon, presumably. Then we do a gel electrophoresis or capillary electrophoresis on it. This will be able to identify how each person has a different one of these areas. We'll detect it. We'll detect it based on the size, and we'll compare the size to that of a suspect. So this might not be clear. Let's show a diagram of how this works. First, we obtain a sample. Now, this is like a swab, whatever it is, a Q-tip that has like cotton swabs on the end. Use that to get the DNA sample. Whether it's from a real sample or whether it's just a suspected place that has the DNA at the crime scene, whatever it is, you pick it up using, let's say, this device, this Q-tip. Then you get it. You have cells. You will have, again, if there's DNA in there, if it actually is a biological sample that contains DNA, it will have cells. You have to rupture the cell and get the DNA. That's that little thing over there. Get it out of the cell. Once you get it out of the cell, you replicate the thing, but you don't replicate everything. You replicate the STRs, the short tandem repeats. Those are different parts of the DNA. You might remember that DNA is composed of billions of base pairs. A group of base pairs, would be the STR, the short tandem repeat. I mean, not all base pairs. It would be short tandem repeats, but we look for the short tandem repeats in those bases. So we get them, we replicate them, and we mentioned the polymerase chain reaction earlier. That's how it works. So we replicate all the important parts of the DNA. We take the DNA from the sample, and we put it through gel electrophoresis or capillary electrophoresis. The basic idea is that you send it through something, and differently sized STRs will go through faster, or will go through first. So let's say this one went through first, and this was the slowest one. However it worked, presumably these are the larger ones go through faster, they go through first. So anyway, so you put your STRs and your sample that, the multiplied STRs that you have, put it on your gel capillary electrophoresis. You'll send it through the detector. You will see a chromatogram or a graph of, where the thing ended up, and this is your DNA profile. So if you have a suspect, let's say at a crime scene, you picked up this DNA, and here's your DNA profile. You can check it with an actual person. Say, oh yeah, is the person who was at the crime scene the suspect? Or what we're seeing here is we have someone left their DNA at the crime scene. We check it with a CODIS. The CODIS is the Combined DNA Index System, or identification system, the index system, the combined DNA index system. It's a database containing DNA profiles from all kinds of people. People who are incarcerated, sometimes people who work in the labs. Some people, they have to have their DNA put in a database. So that's all in the CODIS database. So we have this crime scene DNA profile. We compare it to what's in the CODIS, the database of all DNA. We can find a match, so we know, oh yeah, the person in the CODIS, whatever that guy's name is the one who was at the crime scene. So that's how DNA is used. It's a general scheme for the way DNA is used in forensic analysis. So we mentioned the short tandem repeats. Those are the parts of DNA we want to replicate. Those are the specific things that we're looking at in order to use DNA to identify the person. So what is a short tandem repeat? What is a tandem repeat or a short tandem repeat? We mentioned this in the earlier lecture, so let's just do this briefly. There are parts of the DNA that everybody has that repeat themselves. In this case, it's a repeat of this block. Here you see there are 4 of them. Now, everyone has this in their DNA, and the question is whether it's repeated 4 times, 7 times, 12 times. Every person has it repeated a different number of times. Some people can have it repeated coincidentally the same number of times, but different people have it repeated a different number of times. So we get this area of the DNA that has all these tandem repeats, this tandem repeat. Now there are 16 different tandem repeats that we're looking at. But as for this tandem repeat, again, the question is each person, how many times this is repeated. Different people have repeated different times, so we use that to identify the person. We take several of them, let's say 16 of them, then let's say the first one has the same number of blocks. The second STR, short tandem repeat, was probably not going to have the same as a random person. And if there's if all 16 of them have the same different, all 16 of these different STRs have the same number of repeats between the sample of the crime scene and the suspect, well, that's not a coincidence. We know the suspect, the sample of the crime scene was from this suspect. So we take short tandem repeats, not just any tandem repeat, because the shorter the better. The longer the repeat, the more likely it's going to be damaged somewhere along the long repeat. Smaller, it's less likely they're going to be damaged there. It's also a little bit harder to replicate the larger repeats. When they're larger, it's harder to repeat. There are more because there are more of them. So these are a list of the 16 that are actually used in DNA analysis. I mean, I'm not going to go as to why these codes refer to these tandem repeats, but that's what they are. This is just the name of them if you want to know. And count them. 1, 2, 3, 4. There are 16 of them. Notice this one called XY. It actually designates. It's actually a test for the X and the Y. In other words, the male has an X and a Y. The female has 2 X's. So this will test for the short tandem repeats found on the X and on the Y. Now, if it's a female, you don't have an X. You're going to have 2 Ys. So you're going to actually be, but if it's a male, you're going to see 2 signals, one from the X, one from the Y. Female will only show 1 signal from both X's. So you can identify whether the one who left the DNA was a male or female just by seeing this thing. And we'll take a look at this a little bit more soon. It's again, we try to do 16 of them, all 16. If we only have 13, it's good enough to put in the database, but we try to get all 16. It was mentioned that we put it on a gel electrophoresis or capillary electrophoresis. You don't only have to get this one and put that through it, and then for every 16. Sometimes there are ways of doing 3 or 4 in the same, let's say, gel electrophoresis column. Or sometimes you can even do, there's one that enables you to do all 16 of them on the same column in the same process. So that's called multiplexing. It just makes things a little bit easier if you can do several of them at once. So here's an example of DNA. Of a DNA profile that was obtained. So we're going to say that here's the let's say the electrophoresis column, and would be the time 100, let's say 100 seconds. This is 145 or so seconds. So we see this so whatever it is, the D whatever.